This is a cell, and I'm guessing you already knew that. From the dancing DNA strands, the buzzing organelles, maybe even that massive endoplasmic reticulum stealing the spotlight. Now, cells carry out all kinds of essential functions. But today we're zooming in on just one: how molecules and ions move across the cell membrane, in or out. And if you're enjoying these clear, no fluff breakdowns of cell physiology, hit that subscribe button so you don't miss what's coming next. Let's start simple with passive transport. Passive transport is all about molecules flowing from areas of high concentration to areas of low concentration. Let's begin with the most direct form, simple diffusion. Here, tiny, non-polar molecules like oxygen and carbon dioxide slip straight through the membrane. There are no proteins or channels involved, just movement driven by the gradient. The rate of diffusion is influenced by several important factors. A steeper concentration gradient leads to quicker diffusion. Higher temperatures cause molecules to move more rapidly. A larger surface area of the membrane, such as that found in the lungs, facilitates greater transport. Additionally, smaller molecules tend to move faster than larger, bulkier ones. But remember, diffusion is only efficient over really short distances. That's why capillaries and alveoli are so thin. Now, water follows a slightly different set of rules. This brings us to osmosis, the diffusion of water. Water moves from areas where it's more abundant, meaning low solute, to areas where it's less abundant, high solute. It travels directly or through aquaporins, water-specific channels in the membrane. This simple movement keeps your cells from swelling up and bursting, or shrinking and drying out. All right, but what about molecules that are too big or too charged to pass through alone? That's where facilitated diffusion comes in. Here, your cells provide two types of help: channel proteins, like tunnels for ions to pass through, and carrier proteins, which bind, shift, and release molecules like glucose. It's still passive, still from high to low concentration, and still no energy needed. Now let's head back uphill to active transport. This is where cells do spend energy to move substances from low to high concentration. They're going against the gradient, and that takes work. Let's start with the classic, primary active transport. This is when a protein pump uses ATP directly. The most well-known example is the sodium-potassium pump. It pushes 3 sodium ions out and pulls 2 potassium ions in against their gradients. We covered this in depth in our sodium-potassium pump video. It's a must-watch if you want to understand more about the sodium-potassium pump. Sometimes the cell doesn't spend ATP directly. Instead, it uses energy stored in a gradient. That's secondary active transport. Here's how it works. Primary active transport, usually the sodium-potassium pump, pushes sodium out of the cell. That creates a steep concentration gradient, lots of sodium outside, less inside. Now that gradient becomes energy. Sodium wants to rush back in. And when it does, it drags another molecule along for the ride, like glucose. Or amino acids. This paired movement, where sodium flows in passively and another molecule is pulled in against its gradient, is the core of secondary active transport. You'll see this in action all over your body, especially in the small intestine and the kidneys, where nutrients are absorbed efficiently without using extra ATP. So here's the big picture. Passive transport, no energy, just flow. Primary active transport, energy is spent, gradients are built. Secondary active transport, one gradient fuels another. If you found this breakdown helpful, don't forget to subscribe. We've already got full animations on ion channels, the sodium-potassium pump, and active transport waiting for you.